For this assignment, our group chose to read an article that discusses the research conducted by a team of biologists and neurologists from Leiden University in the Netherlands. The article is called Co-expression patterns between ATN1 and ATXN2 coincide with brain regions affected in Huntington's disease. The article proposes that the relationship between polyQ genes in the brain, genes that contain repeating CAG base sequences, could have an effect on the development of Huntington's disease and other neurodegenerative diseases. This concept is shown here in a couple different diagrams from external sources, and basically they all show repeats of the CAG sequence that form protein aggregates that have harmful effect on neural tissue. Like any scholarly journal, the purpose of this article is to explain the research conducted about a particular topic, in this case, how interactions between genes might affect polyQ diseases. And the goal is to translate the specifics of each experiment to a general scientific audience that can understand maybe scientific concepts and the methods used, but who might not have as an extensive background in this particular field. My figure was figure 1 in the article, and as you can see, it doesn't exactly focus on a single experiment, but rather gives a general overview of the experimenter's approach to the study as a whole. Part A, like I said, gives the general approach conducted in the study, a flowchart of inferences made at the very beginning to larger applications of the results found at the bottom. Part B shows samples of co-expression networks found in HD-associated regions of the brain compared with the rest of the brain. And the second image shows samples found in other anatomical structures of the brain, again compared with the rest of the brain. Section C shows the mapping of the Allen Human Brain Atlas, which maps gene expression in healthy brains. And some of the samples mentioned in Part B are placed in the anatomical structures in and out of the HD-associated region. Images D through F are grouped together because they show the co-expression networks and the links between correlated pairs of genes. And lastly, Part G compares the polyQ relationships in the region associated with Huntington's disease with other structures of the brain. So the results of the experiment are that there are several connections between genes that commonly express the CAG sequence and these genes that have been associated with Huntington's disease and other neurodegenerative diseases before. And the strong co-expression patterns found between specifically the ATN1 and the ATXN2 genes, and the relationship with the HTT gene, which codes for Huntington's. These patterns suggest that the development of Huntington's disease might have to do with these interactions. The results demonstrated in this figure particularly imply that there is a correlation between the location of gene co-expression and the regions that already have established links to HD. The authors can conclude that since these gene networks are found in healthy brain regions associated with HD, they could be affected by the polyQ mutations, and gene expression in a healthy brain could make other regions of the brain susceptible to these mutations and these errors in gene co-expression networks. In the conclusion of the paper, the authors admit that their findings could not really be validated by any other research because not a lot of other research has been conducted. More investigations need to be done in this area. But this study definitely provides the field with a starting point from which other research can begin. Thank you.